So, first of all, I thank the organizers and the International Institute of Physics and the director, Professor Siddhartha, for this invitation. And I will divide my talk in two parts. First, I will talk about electric fish, and the second one is about rattlesnakes. Well, I work at the Institute of Physics in São Carlos, of USP, and in the Laboratory of Neurodynamics and Neurobiophysics. Okay, so I don't know exactly what you mean when you say this history about the Lord of the Rings, but I remember, it is nice because I was planning to talk about this, because this title, Physics and Neuroscience, is like a dream for me, because in 2002, after 2 years in a postdoc in San Diego, I came back to Brazil, and I got employed. And I got employed as a professor of physics at the Institute of Physics of São Paulo. And I was dreaming and continuing the work I started doing in San Diego, interacting computers and living neural tissue, right? And all my background was in physics. I did my PhD with the Dripping-Foster experiment. I was an experimentalist, but working with chaos theory and dynamical systems applied to experiments. And then I went to San Diego and started working with models of neurons and how to make models that we are able to simulate in real time in the computers and connect these models to real living neurons. So that's the idea of computers and living neural tissue in real time. And it was fascinating. But there, we were working with the California lobsters that are very tasty and everything. But back in Brazil, I don't have lobsters. I don't have a laboratory. The only thing I bring with me was an old microscope, like a 74 microscope that my old PI in San Diego gave to me as a gift, and an acquisition board and something like that. Nothing else. And then I start looking for, okay, what I'm going to do now in Brazil. And then I start looking for animals and for experiments that I could bring to the Institute of Physics and start working in things that I like. First of all, I try the crabs, because you have the lobsters that are crustaceans, so the crabs are exactly the same, so I could work with the crabs and continue my work with crabs in Brazil. But also, I have contact in San Diego with some people that work with electric fish, and they were really fascinating, because electric fish, this kind of weakly electric fish, not the enguia that gives you an electric shock and makes you unconscious, but these guys, they generate a very small electric field, only about 2-3 volts of amplitude, and they use this electric field, it's a pulsed electric field, and they use these pulses of electric field to make a kind of spectroscopy of what is around the fish. It's really amazing. So, the fish is there, okay? Something is around the fish. There is an object here, or just the environment, or something like that. So the fish generates a pulse that we call electric organ discharge. That's a pulse that looks pretty much like a spike of a neuron. It has like a one millisecond of duration and 3 volts of amplitude. And this electrical field propagates inside the water, interacts with the object, and goes back to the fish. And the fish analyzes what kind of distortion happened with the propagating electrical field, and based in this distortion, it's able to determine the size, the distance, and properties of what the object is made of only by looking at the distortion of the electrical field. So this is really amazing what these animals can do. How they do that? So this is how the electrical pulses look like. So this is an electrical organ discharge. I have another one here, another one here. This is a Gymnotus carapo that we use here in Brazil. That's very, very common. This animal lives in dark waters and kind of muddy waters. So for them, it's very useful to have this property of looking around without need to use the eyes, because they live in usually dark areas and it's difficult to see. And they are also animals with night habits, so they prefer to go out at night and hunt and so on. So this is a kind of fish that we call pulse fish, because all the pulses have exactly the same amplitude. They are very stereotyped. They don't change very much. They don't change almost anything. The only thing the fish can change is the distance between consecutive pulses. So it looks like little spikes there. And this is how the electrical field lines look like into the water. So the fish is almost like what we call in physics a dipole, okay? And you have these electrical field lines going from the tail to the head. And the head of the fish is full of electrosensors that receive the electrical field propagating to the water and make a kind of electrical image of what is around the fish. So this is how it works. So you have an object, the electrical fields are there. So this is a kind of insulator, you can see, because it makes the lines, electrical field lines, goes more apart. So the thing works like this. You have the pulse here, the electrical field propagates into the water. If you have an object that's metallic or a conductor object, it makes the lines get closer. If you have an object that's an insulator, it makes the line get apart. So this is the way the fish, the same size, same color, same everything, the fish can imagine what's made of. If some insulator or some conductor. And then the fish can decide if this is good to eat or not, for example. So if you look at the density of electroreceptors, starting from the jaw, from the tip of the jaw of the fish and going to the tail here, the fish length, you have most of the electroreceptors are in the region close to the jaw, in the head. And this is so, the concentration of electroreceptors is so bigger in the jaw region that's even called sometimes the electrical fovea of the fish around the mouth. So it's really good for them to have this, because if they are going to decide to eat or not, when the animal is, or the other animal is very close to the mouth, they have the best vision, electrical vision of it, so they can decide if they eat or not. Okay? So, more than that, all of these characteristics that I'm showing here, they are used for this we call electrolocation. That's this kind of a spectroscopy of what's around the fish, and kind of electrical vision. But these fishes also use their pulses, the electrical pulses, for what we call electrocommunication. Yes? More or less. They can. I saw a paper when they made an experiment. They trained some fish after 3 or 4 days to recognize small metallic objects. And the fish were able to, they put like in 2 boxes, one empty box and one box with the object. The fish was able to swim 10 centimeters apart from the boxes. And if the fish swim over the box with the metallic ball, it gets some food. And the fish was able to do that like 99% of the times after training 2-3 days for balls 1 millimeter diameter. So it's really good. It's not bad. 10 centimeters, 1 millimeter. It's really amazing. It's a good. I don't know how we can compare that with vision, but it seems to be very, very good. So they can also recognize the species. They can change the kind of changing their patterns of pulses, changing the duration of the pulses and the time between pulses. They can accelerate or decelerate this rhythm of generation. They can talk to each other, and they can decide things like territory, dominance, and reproduction. I have a friend in the INPA, that's not the Institute of Mathematics, but the Institute of Pesquisa Amazônica, and the Zé Alves Gomes. He has a problem there that's really interesting. The Amazon River is such a barrier for this fish that one fish that is 1 a small river at one side of the Amazon River, it's never able to cross and get to the small river at the other side. So they collect fishes of this species from one side of the river and from the other side of the river. If you look at them genetically, like the DNA of the fish, it's 99.999% equal. The fish look the same, so for a biologist's point of view, they are the same species. They put them together, they don't match. They don't get offsprings. And one hypothesis they have is that they are isolated from one side to the other for so many time that probably their language changed and they are not able to talk to each other anymore. Probably. So if this is true, it's a point where you are in the almost having two different species emerging from a single one. But you only know this after, I don't know how many years. But it's really interesting that they don't get offsprings anymore. Well, so here, all these things made me believe that this work with these fishes could be something really nice because they use this electrical signal for so many things. And it seems to be so easy to measure the signals, and you don't need to put electrodes inside the animal because you can do everything without any influence internally the animal. Look at this. This is a picture of a book. When a physicist sees something like this, look, they'll have a fish here, a small tube, the fish is inside the tube, two pieces of metal, two wires, an amplifier, a sound amplifier, and then an oscilloscope. Everybody has this in like a physics tree laboratory. And then you can record the signals from the fish. So this is really appealing. It's so easy to do the experiments that we decide, okay, let's try to do something with this fish. Here you can see the difference between two different species. The Eigenmania, that's what they call the wave-type fish, that generates like a humming, like a and it modulates the frequency to change, to talk to each other. So they have a mean frequency, and they modulate around this frequency to put some information. So in these guys, the amplitude is more or less constant, and they change the frequency. And then you have the gymnotus, that's the pulse type, that changes the interpulse intervals, very, very much from one to the other. Okay. So, question? Exactly. This looks like FM, and this is digital, something like that. So for us, for acquisition, for acquiring data for experimentalists, it's easier to work with this guy than this, because you don't need to have all the waves recorded. Here you can just have the timestamp, right? If you know these times, you have all the information you need. Exactly like we do with the spike trains of neurons. Okay, so in 2002, my dream was to, okay, let's see what people are doing with this fish. For example, for the pulse-type fish. Usually, you will find papers like people saying, okay, this is the frequency of firing of the electric organ discharge of the fish during one day. So they take the fish from one tank, they put in a small aquarium, put electrodes there, and record in the oscilloscope for 2-3 minutes. Then they write the average frequency, put the fish back. After 1 hour, they go there, take the fish, put the fish back in the experiment tank, measure the frequency again, and do a graphics of frequency as a function of time. But every time they go there, pick up the fish, put here, and so nobody was trying to do some really huge data acquisition for, let's put the fish there and record everything and see what happens. Or even better, if you can put more than one fish and see what's going on. Nobody was trying to do this because the experience was difficult, fish moves a lot, and so on. And they try to immobilize the fish. Usually they put a net or something to keep the fish close to the wires they use to do the recording. So, because you have like a dipole sensor here, positive and negative, and the fish aligns along the dipole, you have the maximum amplitude of the pulse. But if the fish moves like this, you lose the signal. So you need to put another set of electrodes and so on. That's the idea. So most of the people used to put a net around the fish and restrict the movements of the fish. So, in my point of view that I'm a physicist, this was very far from what the fish could be doing in nature, when there are no nets, nobody trying to put wires and things like that. So, very naively, we try to do the first experiment with the fish. Why am I telling this? Because first, we don't have the fishes there. We tried to contact people at the Institute of Biology, and we had no success and so on. Finally, a friend of us went to a store where they sell fish and stuff for fishermen. One of the main applications of this fish, the Gymnotus carapo, is as bait for other bigger fishes. And they are very useful because they breathe air, so they take the fish, put in a can or something with a little water, and leave the fish there for 2-3 days, and the fish survives. It's really amazing. Then they catch the fish, take pieces, and put in the hook and try to pick up bigger fish. So if you do this, you can go to the store, buy as many Gymnotus carapo you want, and go fishing, no problem. But if you want to take one of these fish to your laboratory in an aquarium and put 2 electrodes there and record the fish without interfering with his behavior and giving food to him and everything, you can't do that. Because you need the authorization of the IBAMA, you need to go to, it's really amazing. It's really amazing. You know this. It's really amazing. Right? You need yeah, but if you don't ask, you can, no, it's even worse. If they catch you with a basket in your car, and the police officer come and, what's this? Oh, it's a fish, and what's it for? If you say, it's bait for fishermen, oh, it's okay. But if you say that you are going to do experiment, you go to the jail. Because it's animal silvestre, right? It's yeah, it's murder, exactly. And then when you go to the IBAMA website, you discover that you have to include the PDF of the report of the ethics committee that analyzed your experimental procedures with the fish. Okay, then you go to the website of the committee that analyzes, a committee of ethics, and they ask you for the number of the IBAMA authorization. How do you do that? Huh? So the way we figured out to do that was we told both of them that the other one was doing. And then we got a report from one of them of, okay, it's positive for us, but you need the number of the IBAMA. And the IBAMA said, okay, it's positive to us, but you need the report of the ethics committee. Okay, I put 2 of them together and that's it. But if not, you can't do that. You go there and get stuck, Well, so we smuggle a fish into the laboratory somehow. And then we try to do that in the simplest, based in that picture, right? In this picture here. We try to do the first experiments in the easiest way possible. We try to use a single differential amplifier. So that means I have 2 wires. But I want to let the fish free to move around the aquarium as you wish. So what we did, we choose a cubic aquarium like this. And then we put an electrode in each vertex of the cube. The problem is that I have 1, 2, 3, 4, 8 electrodes. The electrodes were very, very easy to do because usually the fish tanks, they are made of glass and this silicone glue, right? And the silicone glue is really cool because you get one of these stainless steel wires that people use for odontometric purposes, for prostheses. And then you stick the wire through the silicone, and it gets completely tight, and it never leaks any water. It's really, really cool. So we use these methods to put little wires here, and these were our electrodes. But then, how to turn these 8 electrodes in only 2 wires? The way we very smart tried to do that was like this. Okay, let's put together diametrically opposite electrodes, like this. This guy together with this guy. Then, this guy together with this other guy. This one with that one, and this one with that one. And then, now we have 4. We reduce it to 4. And then what we did? Okay, this one is linked to this. And this one is linked to that. So this is going to be the positive, and this is going to be the negative. And then we did the same thing with the other 2. Okay? So we put everything together in just 2 wires. So this way, it doesn't matter with the position of the fish inside the tank. Always that there was an electric pulse, we are able to catch it. Okay, the problem is the fish is used to swim into the water where there are no electrodes. And there are no short-circuited electrodes like that, right? So the first thing the fish did when we put it inside this tank was start biting all the electrodes as much as he could. Okay? He got so upset, but so upset. It was amazing. It took like 3 hours for the fish to get used to the electrodes and stop biting them. Okay? After that, we found many other problems, right? For example, if you turn on, after the fish get used to the electrodes, we did the same thing as you see in the book, right? We put oscilloscope, we put the audio amplifier to listen the fish, and you could see like you could hear the noise like of the pulses. But then, if you turn on the light or turn off the light or go close to the aquarium, every movement you do, the fish was accelerating. Like, fish was like this. Somebody opens the door. Turn on the light. There was a bus stop in the front of Institute of Physics that time. You remember Rua do Matão? And the buses, I think old buses, there is a speed bump or something like that. The bus need to brake very strongly after going down in a hill. So you could listen the bus doing like this. And the speed bump, boom, and the fish. So it was really amazing. If we want to record that and relate this to anything, I don't know, for the fish it was really easy to record, only 2 wires, but to record all the other sounds, vibration, light, and everything that was going on, we got completely lost. So, lots of problems, we gave up, okay? Okay, it's not going to be like in the book. It's not just put the 2 wires there and so on. Well, what we did then was start thinking about a serious equipment for recording these animals. And at this time, it was nice, because of this Lord of the Rings thing that I remember, I was invited. I submitted a work to the National Meeting of Physics of the Brazilian Physics Society. And then we went there. I showed them something about a poster about the fish. I showed a poster about the neurons and so on. And people came to my poster and, oh, Reynaldo, I remember you used to work with the dripping faucet. Yeah. But now you're not physicist anymore. What you are doing? You have animals. You have fish. You are opening crabs. Lots of people came to the poster, but more for the curiosity than for the people looking, say, are you okay? Are you sure you're okay? And then, sometime after that, a friend in the Institute of Mathematics and Statistics asked me to give a talk and say something about modeling using dynamical systems of neurons and so on. And then I went there. So the physicists start calling me biologist because I was not physicist anymore because I deal with animals and things like that. And then I went to the Mathematics Institute, I gave my talk, and then a guy at the end told me, oh, you should be a biologist because you were not using any rigor, you didn't improve anything, you don't know if your data are not enough, you don't know anything about noise and blah, blah. So it's like that, right? The biologists used to say that I was a mathematician because I use equations. The physicists say that I was a biologist because I use animals. And the mathematicians say I was nothing because I was not rigorous at all. So it was like nobody's land. Yes. So then we decided to do the evolution to our first serious apparatus. What we tried to do was to isolate completely the environment of the fish from all kinds of perturbations we could have. So this is the tank, the original tank of the fish, with those electrodes and everything. And then we put this tank inside the wood cover, and with a layer of foam, and insulate also by air. So this inside box was hanging with steel cables from another box, also insulated with foam, and the whole thing was hanging from the ceiling. Okay, this was the best way we found to remove vibrations. This was something that I learned in my PhD when I was trying to do the experiments with the dripping faucet that was also very sensitive to vibrations. So we found that hanging heavy things with the stainless steel cables is the best thing to remove those slow frequency vibrations. And of course, with aluminum sheet here to make like a Faraday cage. So you have a double Faraday cage here, one here and another one at the side. So, and this tank was like 40 centimeters by 40 by 40, about 60 liters of water. And our acquisition system, now we decide to leave all electrodes insulated from each other, so we use a differential amplifier with a high impedance of input. We use one of the electrodes as a reference, and all the others are amplified against this one, okay, and recorded in a computer for acquisition. We use the second computer to generate a stimulus. What was the stimulus? Previously, we recorded the wave of the fish during one electric organ discharge, like we record like 5 or 10 or 50 of them. We take an average of them and store this in the memory of the computer. So we know how does it look like the waveform of a fish during a spike. Okay? Then we could choose in this computer any kind of inter-spike distributions. Okay? Random from a real fish, periodic, whatever you want. And then when in the right time, the computer generates, using a digital to analog converter, a signal in a dipole that mimics the real waveform of the fish. And we put this dipole inside the PVC tube, close it at both sides, so for the fish, it was as if there was another fish inside the tube. Okay? That was the idea. The problem we found was, if you look at each electrode, depending on the position of the fish, you get a different form of wave. Of course, because depending on the position of the dipole generating into the water, you have different signal in each one. So at first, this looked like a problem. But then at the end, it turns out to be a good thing for us, because you can detect other features from these different signals. So the software of acquisition was something like this. We have a stimulus that we call S, and you have 7 electrodes, 7 signals coming from the electrodes of the tank, right? these electrodes contained both the stimulus signal that I generate artificially and the real spikes of the fish, the real electric organ discharges of the fish. But since I am generating the stimulus, I also have the general, the signal, the pure stimulus separated from the signal of the fish. So inside the computer, what we could do, we just sum up everything coming from each of the electrodes. We sum the square of the signals just to make them positive. And you have something like this. You have a bump here. This is a stimulus. This is real fish. This is a stimulus squared. This is real fish. And this is a stimulus. Then we do the same. We square the stimulus signal alone. We put a delay because of the electronics of detection and everything to make this minus stimulus square happen in the same time as the stimulus square of the signal that I'm detecting in the electrodes. And then we sum both of them. When you do that, we can cancel, very carefully, you can cancel the stimulus from both signals. And then you get only the fish signals. Okay? Then you have like a threshold detector, very simple, and we can record only the spike time. Moreover, since we have the signal in each one of the electrodes, we started recording the local induced amplitude, the maximum and minimum. For example, in electrode 1, you have the pulse of the fish. One pulse was this. It reached this amplitude positive and this amplitude negative. The next pulse was like this. The next was like that. What can we predict looking at this amplitude? Here, the fish was closer to be aligned to the electrode 1. And here, it gets more far from that electrode. In the electrode 2, it happens the opposite. At the beginning, it was not so close, and it get closer as the time passed. So we could use these amplitudes, different amplitudes that at the beginning looked like a problem, as a way to infer the fish position inside the tank, only based in the electrical signals, without needing to record from cameras or something like that. The idea is this. So these are the 7 electrodes, and the pulses maximum and minimum each one. You look at the electrode that has the highest amplitude, this one, that's electrode 4, and then you say, okay, if the positive amplitude in the highest amplitude electrode is the biggest one, the positive amplitude is bigger than the negative, means that the head of the fish is closer to the electrode. If it's the negative, means that the tail is close to the electrode, because the head is positive and the tail is negative in this animal. And then you put a red ball there close to the electrode 4. Okay? So that's the idea. So just to show how it works, very, 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 very simple. Only using the highest positive amplitude electrode. I think that we use the first and the second one. Only 2 of them. And just a linear model. Okay. So this is the tank, the experimental tank. You can see the fish is here. Okay. The electrodes are in each one of the corners here. And this is the artificial fish. So it's the PVC pipe, the electrode, artificial electrode for stimulation is inside here. This is the fish, and this is the computer-generated movie of where the fish should be. Red is the head, and black is the tail. So let's see. You see that's not very, very good, but most of the time works. The fish is here now. We just divided the cubic aquarium in 8 regions, so it's very rough. We believe that using support vector machines and machine learning, you can do this much better than that. with the same signal. Okay, so here the fish is not being stimulated, so it's just exploring. We just put it in the aquarium, so it starts exploring and seeing the new environment and so on. These fishes have names now. And there is a technician that feed them every day and so on. But unfortunately, IBAMA, there is an obligation of us to sacrifice them every 12 months. We cannot stay with them, even if we don't do anything with the fish. We cannot put them back in the nature or something like that. And we cannot keep them in the laboratory for more than 12 months. Okay, so it started again. Okay. So, these were the first nice data we got from our experiment. So this is what we call time histogram of the interpulse intervals. I think most of you are used to this. It's very simple. So each one of these vertical lines represents an histogram that was in a window of time. And you see in red the highest probability of finding an electric organ discharge, the time between two of them. So the fish starts firing at like 30 milliseconds of interval between them. So it's very, very, very fast. And after 8-10 hours, it starts relaxing and showing regions where you have much lower activity, like 45 milliseconds, and then changing from 45 to 20 or someone. And here, you can see very long times when the fish is about 45 milliseconds and so on. And this other graph, so this is 48 hours, 2 days. So first thing we found here was, as soon as you put the fish in a new environment, the fish gets stressed and gets in some looking around behavior to get acquainted to the new aquarium and so on. And it does this for about 10 hours. Okay, so it's a transient behavior of 10 hours. That means that taking the fish from one tank, putting in another tank to making a measure of the frequency and putting the fish back every 10 minutes, you never measure anything related to the real behavior of the fish, because the fish will never get in this region here after 24 hours or something like that, right? Second thing we could see was also nice, that since we have these amplitudes here in each electrode, we have these maximum and minimum values. We can use a window of 1 second or something like that and look how much this maximum and minimum amplitude varies into this window. Or, for example, half a second or 100 milliseconds or so on. You can take the standard deviation of these amplitudes. If the standard deviation of these amplitudes, maximum and minimum, is like almost zero, means you have no variation. If you have no variation in all of them, means the fish is stopped. The fish is not moving. If the fish moves, you change the amplitudes in the different electrodes. So we have an idea to calculate in small windows the standard deviation of these values, maximum and minimum, and sum all of them. So you have something like the sum of all deviations of these amplitudes. And that's somehow related to the movement. If this is almost zero, you have no movement. If this is something really big, the fish is moving a lot. Okay? And this is what we show here, this movement in arbitrary units. So at the beginning, when the fish is firing like crazy, very high frequency, you can see that the movement is really high. Right? The fish is looking around all the time. And then after like 8 hours, the fish starts jumping from regions where there are almost no movement and a lot of movement, almost no movement and lots of movement, and so on. Okay? And here, I show two different windows here. One at the beginning of the experiment, and another one after, in the middle of the second day, to show the difference. Here, you have the interpulse intervals, the histograms in the time, and you have either the movement and also the entropy of the electrical signal. What's the entropy of the electrical signal? If the fish is spiking like this, tech, 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 periodic, the entropy of the signal is 0. There is no entropy. If the fish is doing something like this, tech, 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 this is a lot of entropy. So we calculate the entropy of the signal. I will show later how we did that. But the main idea is this, is to see how much the electrical signal is able to vary in the time, okay? And you can see in the beginning, the entropy is low, it's really low. Why? Because the fish is firing almost periodically, but very high frequency, right? At the end of the experiment, you have regions where the entropy is lower and higher, lower and higher. And these regions coincide with the movement of the fish. So when the fish is not moving, you have almost no entropy, the frequency is low, and the entropy is also low. We believe in these regions, the fish is sleeping. The fish is not doing anything. It's just stopped. It's not trying to explore the environment, and it's not doing anything. You have low entropy, you have low frequency, and you have no movement. So this is a way of just looking at the electrical signal of the fish, we can predict if the fish is awake or sleeping. Okay? And we have some dreams of working with this and Siddhartha to see if we can do something with this fish to see if they learn when we let them sleep or not, and things like that. So these are some ideas. You have any questions so far? Okay.
What is the inter-animal variability in this sort of behavior? Do you have fish that have very different personalities, or are they more homogeneous than mammals?
We did this experiment with 12 different fish. They all behave the same way. Because, I mean, you find the same regions, the same transition, the same like 10 hours, some of them 8 hours, some of other fish 12 hours to get to the more relaxed behavior, but all of them behave the same way. But if you look at the signal of each one of them, if you look at the interspiking interval distribution here, each one of them is different. Some of them are bimodal, some of them is unimodal. But when, exactly, yes. And the same animal, when submitted to the same experiment after 15 days, you can also have a different distribution. Yes, yes. It depends on the temperature, it depends on lots of things. But the average behavior is the same. You have this 10 hours time for... both things. This first experiment, I think this is the first experiment I'm showing here. This one was in the dark. And then we replace it by another one when you have a sensor of light that turns on a light inside the aquarium when you have sunlight outside. So you have the circadian rhythm maintained into the aquarium. Okay. Practically the same. The only thing that you see here, here the fish was in the dark. But you can see that here the fish has more episodes of this kind of sleeping-like behavior. And then again here. The distance between this region and this is about 24 hours. So this is like a circadian rhythm. So you see lots of activity here. Probably this is the night for the fish, because it's more active at night. And this is night again, and this is day. It sleeps during the day. When you turn on the light, you intensify these episodes of sleeping here. You get them first and more easy to detect. Sure. Yes, all the time. It's like that. We don't know. They are able to stop firing. They can do that, but they do it consciously. Usually when they are put together with another fish of the same species, a biggest one, a bigger one or something, you put them together, and then they start fighting with electrical signals, like the electrical communication. One of them, and if the other one realizes that this one is much bigger than him, it stops firing. It don't want to be detected. It tries to hide. And it can stop for 10 seconds. It's really amazing. Okay? And it's cool because a friend, Angel Caputi from Uruguay, he has an experiment when they record chronically implanted these fishes, and they show that even though the fish is not firing, the pacemaker is firing inside. So it's an inhibition after the pacemaker. Because this fish, this electrical organ discharge is generated, I forgot to say that, but generated by modified muscle cells. They are not, what generates the electrical generation is not made by things like neurons, it's muscle cells. And you can inhibit them with curare. But it's really nasty experiment, right? You need to, it's really bad because what they do is they curarize the fish, so the fish is not able to move, but the fish feels everything, thinks everything because it's not anesthetized, right? It's just unable to move. All the muscles are stopped. Then you need to force the ventilation because the fish is not going to breathe and it dies. So you need to put a tube and do forcing. So it's very bad experiments with curare. But the generation is like that. But you can see that even when the fish decides by itself not spiking, you have the pacemaker working and the inhibition is after that. Is a little bit before the motor output. Okay, and these are our first experiments with stimulation. So here we have epochs of 30 minutes. First, we have 30 minutes without stimulation. Then we stimulate with the same, look, the signal we send to the artificial fish is exactly the same. Oops. Wow. No. It closed. Yeah. Okay. So the signal we send to the dipole inside the plastic tube, the artificial fish, is exactly the same here and here. The only thing that changes from one case to the other is what we use as the distribution between interspike intervals. The spikes are exactly the same, pre-recorded from the fish and so on. Okay? So here, we pre-recorded the interspike intervals of a real fish. So we go there, we measure the interspike intervals, and then we reproduce exactly the same thing. So it was like this pre-recorded fish was inside the plastic tube. Exactly. But it was not able to listen anything. It was just speaking. Okay? Just a playback. The bigger one, the smaller one, we tried a lot of them. And all of them have the same behavior here. Like lots of activity and changing activity. Lots of movement and lots of entropy. But when we took the same distribution of electric organ discharge, the same series, the playback, but then we randomly scramble the interspike intervals. So I take interval here, this interval 10 was the first, the 96 was the second, and so on, randomly choosing the intervals. We got this behavior. And you can see that here the fish is sleeping. Yes, it was sleeping before. Then when we start playing, it woke up. Then it got a little upset or so on, some activity, and then after a while we start sleeping again. So it looks like the fish is able to discriminate between what's real, you can see that here you have for like, I don't know, 10 minutes, you have some activity and then you go slow it down. And here it never happens. And this happens for all the playbacks we tried. Bigger fish, smaller fish, and so on. When you scramble the data and play to the fish just, at the beginning, gets upset and go there and try to bite the tube and so on, start swimming around. After a while, we just gave up and go sleep. But we also have something like when you have this behavior like this here, no sleeping, and then we turn on the stimuli with random. And then you have the same thing. You have an increasing of activity at the beginning, and then it goes down and eventually sleeps. Not all the fishes sleep, but some of them. But they never sleep. The interesting is this. Some of the fish even sleep for the random order of electric organ discharge intervals. But no fish sleep for the real order. But this kind of behavior, when the fish somewhere starts sleeping or stop paying attention, it happens even if you change the order. Like first you play the random, and then after a while you play the real. Because this is a problem also you have, right? Because if you take here the real signal and then the random, maybe the second epoch of stimulation, the fish was already used to here, and say, okay, nothing gonna happen here. So we try to do with new fish. We try to do this when the fish came to the laboratory in the first day, after a week, after 2 weeks, after a month, and so on. We have all of this, lots of data. And nevertheless, the way they react inside this epoch of stimulation is different. And it's really amazing because if you look at the signal of the interspike intervals as a function of time, they are also different for the same fish for different presentations of the same signal, and also between fish. But then we did some analysis using information theory. When we found peaks of average mutual information between stimulus and response of the fish, in different fish, even though this entropy was different, the movement was different, and the signals of the both were different. So something is there. We don't know exactly what, but it looks like something is there. Okay? But it's just preliminary, it's not conclusive yet. Okay. So then, as I said, this fish is very territorialist. They like to fight and so on. And another thing that we like to do was to have a big tank, put 2 fish inside the tank, record the signals of the fish, of both of the fish, know the positions of the fish, and relate the electrical sequence of pulses with the behavior of the animals. So that's our dream. The problem is that we have something that we call jamming. That means both of the fish tried to spike an electric organ discharge exactly at the same time or very close. Then you have a big problem of detection because who is this signal from? It's the sum of both of them, right? It's a complicated problem. Yes. No, in a camera, and you have an electrical signal, so you don't know which.
You know which one is closer to this electrode.
Oh, yes, yes. And then you need to do something by smoothing, right? So the guy is close to this electrode, is firing, so that's what we try to do. At this time, Caroline was an undergrad student. I asked her to do by hand, using camera and splitting the spikes and discriminating between fish. Well, she tried to do that for 10 minutes or something. I think it was 10 minutes. And then she asked me that she was looking for another advisor or something. And then I gave up. So, okay, let's do something else. So they try to do this handmade spike sorting, but it's a really huge amount of work. And when you have both of them together, you need to start guessing because you never know exactly the position. Now we are doing this, we start doing this automatically using support vector machine and machine learning and things like that. Well, and this jamming issue is also nice because most of the people at this time, and still believe, people that work with electrical fish, that there is something called JAR. That's the jamming avoidance response. Especially because most of the people start to work, like one of the more famous guys that work with light fish, work with the wave-type fish. That was Bullock. And the wave-type fish, like Agemania, remember that it has an average mean frequency, and then it modulates the frequencies, an FM fish. If you have two FM fishes close in space and with the same frequency, you lose completely the ability of doing any electrolocation, because all of the electrical fields will get messed up. None of the fish will be able to recognize anything. Then what happens in those kind of fish is that, I don't know how they do that, but they get close. For example, I suppose both of them are about 800 hertz firing. So if you listen, it's like something like both of them. As soon as they get closer, the dominant fish increases the frequency a little bit. It goes from 800 to 850, and the other one decreases a little bit. Always the dominant increases and the other one drops a little bit. Then they go to another frequency. There are like FM stations, right? They can modulate inside some range. They can survive together and nobody bothers with anybody else. Everything is fine. So what people believed for the pulse-type fish was that, okay, two of these pulse-type fishes firing at the same time is bad. They are not going to be able to do electrolocation. So they have a mechanism to avoid firing together. And they never fire together. It's very hard for them to fire together. The first experiment I did, I put the fish together. It's like 2-3% of the time they fire together. They don't look like avoiding. If you look at the spikes coming closer, they came closer, then fire together 3-4 times, and then they get apart. Then they come close together again, fire together, and then come apart again. It looks more like a beating by chance than something to avoid. If you have some mechanisms to avoid, you have something like this. They get closer, and then they get apart. Closer and get apart. They never fire together. They simply look like ignorating another one. They just pass through. So for us, it looks like this jamming maybe is not working, or this mechanism does not exist for this kind of fish. Something else is going on there. Okay, so then what we decide to do? Putting two of the fish in the same tank was very difficult because they fight, they fire together, we cannot discriminate between fishes. So we decided to replicate the whole apparatus to do another tank with another stimulation, and then we made a software that was able to detect the signal of firing of this real fish, and as soon as we detect, it generates an artificial pulse in the tube, the plastic tube of the other fish. And when the other fish fires, the computer was also able to detect and generate an artificial pulse in the first one. So this is what Siddhartha was calling the Skype between fishes. So for this fish, was as if this guy was here. And for that guy, was as if this guy was here. And in this case, is in both ways. Okay? So both of them will change their distributions of spikes, and the other one, you know that. Okay? The only problem is that the amplitude is fixed. So it's like if there was a immobilized fish inside the tube or something like that. Okay? It's a little artificial, but it's something that is. What we do is, when you have a jamming of both signals, when you get confused, the computer, the SVM cannot discriminate between them. What you do is like this. The previous pulse was also jammed? No. Okay, so let's take the signals in these electrodes in the previous pulse and make a linear combination of them to generate the pulse now. And then we move both of them and choose the minimum error for the fitting. And then we choose the time of each one of them fired. That's exactly what we are doing. But it takes a little time in the computer. So you're not able to do that in real time yet, but almost there. Okay. So here is more or less how this electronics work. So you have one tank here. We square the signal, we invert the signal, and sum all of them. So this now we just want to know when the fish spiked, when you have the electric organ discharge. So we square the signals, we invert and sum up everything, and put this into the computer in one analog input. Then you have the other tank, the signals of the other tank coming here. Also, they are squared, they are summed, and they are fed to the analog input 1. So you have the computer reading analog input 1, 0, and 1 with signals of the fish and the stimulus. Then, this is made by hardware. Okay, this amplification and summing the signal is hardware. From here, we have the software layer. We simply sum the signal that we are just acquired with the signal that the stimulus generated to remove the stimulus from the signal. Then we use the level detector, a threshold detector, to know the time where the fish, one of the fish was firing. And then we record this, we send this stimulus to the other tank and record this time. Okay? It's exactly the same thing what we did before, but now it's double. Okay? We also save for all the pulses the maximum and mean amplitudes at each electrode to know the position of the fish and the movement and this kind of things. Okay, so now I'm going to show a little bit what results we got from these classical information theoretical techniques, like calculating the entropy. So I don't know if I don't think I need to explain exactly how to calculate the entropy, why is like this, right? But the idea is the entropy is the amount of variation that you have in a signal. The entropy is 0 if you have a periodic signal. And we also use the information of a signal. The information that you have of two events, right? You have one event that is S, that's our stimulus, for example, and another event that is this small r, that's the response. And you have the probabilities of finding a stimulus and the probabilities of finding a response. And you have the probability of finding both of them together. So what does this division here means is that if the probability of finding two things together is different of the probability of find each one of them separated, multiplied by each other, means that you have more information in the pair that you have in them isolated. Okay, is it clear? For example, if you go to the cafeteria, and then you have a girl there, and you have a P of S to having the girl alone in the cafeteria. And then you have a boy there with the probability P of R. Boy is the R. Okay? What's the probability if the boy and the girl are not related at all? What's the probability of finding both of them together? It's just the probability of finding one of them times the probability of finding the other one. Right? So, these things should be exactly the same if they are not correlated, not related. If there is a relationship or something going on, the probability of finding them together is bigger than finding them alone multiplied by each other. Okay? So that's the idea of finding the information of two events. And then you have the average mutual information. That means you take this probability of finding both of them, use it as a weight, and then you sum up over all the events, all pairs of events you can have. This is the average mutual information you have. Or you can simply understand that as the average mutual information between a stimulus and the response is the amount of variation you have in the response, the total amount of variation you have in the response, minus the variation you have in the response when you fix the stimulus. This is the noise, right? If you're looking for information between stimulus and response, you fix the stimulus. The amount of variation you have in the response when you fix the stimulus is the noise of the response. If you take the noise of the response given the stimulus out of the all variation of the response, you suppose that that you get is exactly, you get the information that you have from the stimulus in the response. But look here, it's important thing to see that this is symmetric. You can do the same thing using changing S by R here. And that means that there is no causality between stimulus and response. It's just pair of events. You don't know if the stimulus is what caused the response or is the opposite, okay? So one way we try to put some causality there is to use a delay. Like, for example, you have the signal of one fish, the firing of one fish. You have the firing of the other fish, and then you detect the spikes here. Okay? So wherever you have a spike, you put a 0 in your string of bits. And when you don't have spikes, you put zeros. Okay? So this is the way to binarize your series. Then you choose size of word, for example, here 8 bits, and you call this stimulus. You use a delay that is distance between the occurrence of the stimulus and the occurrence of the response. If the delay is 0, both of them are together. You have no causality. But if the delay is bigger than the duration of your word, you know that the stimulus happened before the response, and then you have causality. Okay? But it's something that you have to put by hand in this method. So then, this is what I'm going to show here, is the average mutual information divided by the entropy of the response. That means how much of the variation of the response is being used to code something about the stimulus. And this is percent. So this goes from 14 to 22%. Okay? And here you have two different fish and the same stimulus, the same playback of stimulus. And we played with the amplitude of the stimulus from 1 to 5 volts during time. So not only the interspike intervals were those recorded, pre-recorded from a real fish, but also the amplitude was varying like showing here, from 1 to 5, then drops to 1, then goes to 5 again, then gets stuck in 1, and so on. Okay? And what we found was that during these 10 minutes of experiments, we have regions here in green where both of the fish presented average mutual information of some amount in the same regions. Here in the yellow. In the black, you have a negative result, like you have a lack of average mutual information, and here in red again. But when you look at those signals of this fish and that, they don't look anything alike. Only looking at the signals, you cannot tell what's going on. Jump it here because of the time, was that this colleague of us, Caputi, he has found some cells in the brain of the fish, in the electrical lobe of the fish, that are dedicated to detect when the signal that reached the electroreceptors was fired by their own fish. They are called spherical cells. They did some implant, some chronic implant, and record local field potentials of those cells, and they tend to fire more reliably to their own generated signal than to the signal generated by the other fish. And those cells have a refractory time about 10 milliseconds. So if you recruit all of them and then they fire, they say, okay, you fired it now. You are receiving the signal you generated. Then they get completely deaf for 10 milliseconds. They don't detect anything else. After that, they get sensitive again, and they are able to detect again another spike. But they have this refractory time of 10 milliseconds. And this is nice. This is something that you are trying, they did there with the fish implanted, but the fish was not able to move. The fish was tied in the net, anesthetized, and things like that. We want to do some experiments with this fish, electronic implanted, and sending this signal to us by FM modulation inside the water. And do the experiments with the living fish implanted inside the water, wireless. this is something really cool that we want to try to see if we find the same results when the fish is free to move. Okay. So in the same idea of this refractory time, we have this experience with the artificial stimulation. I'm going to show you two different histograms. One that is called the PST, the post-stimulus time histogram, and the post-response time histogram. What's the difference between them? The post-stimulus is the time after a stimulus was given to the next spike of the fish. Okay? So you have a stimulus, you start the chronometer, and then you stop it when the fish fired. Okay? So this is the post-stimulus time histogram. And then you have the post-response. That's the opposite. You look when the fish fired, and then you get the time until the next stimulus happens. That's the post-response time histogram. Okay? This is very interesting. If you take the real distribution of interspike intervals of a real fish, you have something like this. This is 10 milliseconds, 15, 20 milliseconds is almost the time for the fish to fire again. So that's why it drops. Okay? So you have something like decreases a little bit here and increases a little bit here. But if you have the same distribution from the real fish, but scrambled, okay, ordered, random order, you get this thing pretty flat. No change at all. And then we did this for 10 fish, we normalized it and so on, and this is what we found. So this is the post-stimulus time histogram. So in between 0 and 6, 7, 8 milliseconds, you have a decrease of the probability of the fish to fire. And then you have an increase of the probability of the fish to fire between 8 and 12 milliseconds here. And in the other direction, the post-response time histogram, you have an increase from 0 to 8, 9 milliseconds and a decrease about 10. What does it mean? If we believe that our fish, the Gymnotus carapo, has the same kind of spherical cells with refractory time, this gray region should be more or less the refractory time of the fish. So, post-stimulus, the fish that is interacting with me just fired. If it has a refractory time, I know that it's not going to listen me from here until here, more or less. Then if I want to be listened, shall I spike here or shall I wait a little bit and spike later? So I prefer to spike later. Conversely, if I just spiked here, the post-response, what happens? You see that the increase of the spiking of the stimulus is in this region, in my refractory time. So, what we could figure out from that? When you have periodic stimulation, like Nogueira Caput did, or real easy distribution, if you do something like a sinusoidal wave modulation of the interspike intervals, this is periodic. The fish it's predictable, and the fish does exactly like that. Change the histograms to fire more after the refractory time and fire before the refractory time of the other one. But if the signal is random, is unpredictable, then you get the flat distribution. The fish doesn't know what to do. So what we believe now is that it's not that the fish is able to know that the distribution is coming from another fish, but he knows that he can predict when it's going to be the next spike. And he can do that from a distribution from a real fish or for periodic distributions, but he cannot do that for a random one. That's why it failed and gave us the flat distribution. Okay? And it does that for trying to fire when the other one is going to detect its firing. And D tries to move his firing rate in order to avoid the firing of the other one. Well, this is very different of the jamming avoidance response. This is another mechanism, but we don't know yet. This is just preliminary results. We need more data of two fish interacting. Skype data or experiments that we're planning to do with both fish in the same tank and so on. Okay? So when you have two fish interacting, the post-stimulus time histograms, they become like this. They become like bimodal. You have like both of those. Remember that one was like this, the PSTH was like this, and the PRTH was like that. When you put two fish together, both of them are doing the same thing. They try to do. Then intuitively, I can imagine, okay, maybe the response of all of this will be the superposition of both of them. And I would expect a bimodal distribution. That's more or less what we can, with a little goodwill, see there. Right? And this is interesting because, just speculation now. If this is true, if you have something about 10 milliseconds that the fish doesn't see what's happening, that means 2/3 of the electric organ discharge of a fish falls into the refractory period. And they are not detected. So all of those spikes that I was putting one there in my string of bits, 2/3 of them are just noise, are just things that are not doing anything, right? And if you consider that only 1/3 of them are really useful, we can do some prediction. For example, let's suppose the fish is firing at 40 hertz. That means roughly 40 bits of information per second, right? One third of this is about 13 bits of second. Let's suppose this is used for communication. If we take our an alphabet of 23 symbols, we need 5 bits to code those 23 symbols. For the fish, we have something about 3 symbols per second, right? 13 bits per second divided by 5, about 3 symbols per second. A speaking human, speaking slowly, use usually 11 symbols per second. So if only 1/3 of the spikes of the fish is used for communication, and we assume, that's really crazy, that the fish uses 23 symbols like our alphabet to communicate with each other, the speed of communication is about the same as in human beings. It's really crazy, this result. But it's just speculation, right? And this is the new apparatus we are planning to, we are already started building. So it's a big tank, 1 meter by 50 by 50 centimeters. We have now 12 electrodes, all of them differentially amplified and so on. And we are able to detect the fish position from camera images. This is very easy. We use background subtraction, histograms, and I have a student there working with Python and so on that can do this very quickly. And we even try to use the Microsoft Kinect to do that because it gets so famous, right? Everybody's using the Kinect and so on. The first experiments we did were pretty positive, like you could see the fish in the infrared image, but when we assemble everything, when we did the right thing and to see if it worked, the problem is the skin of the fish is a very poor infrared reflector. It absorbs almost everything. So you can only detect the fish until it gets 10 centimeters deep into the water. If it goes a little deeper, then you get dark and don't see anything else anymore. So we gave up using the kinetic, and then we're going to use 2 cameras as usually. But now we're going to put the cameras over the aquarium and not one over and one here, because my student wants to do some method also to do these experiments in field. So we want to go to a small lake and have the fish there and put the cameras there and be able to detect the fish just from above the lake. What else? now we are doing the discrimination of fish using clustering algorithms, machine learning, support vector machine, and we use FFT and wavelet components to separate the signals. And after training, you put one fish first and say, okay, this is fish A, and let the algorithm learn how does fish A behave. And take the fish A, put fish B, the algorithm learns how is fish B, because the electric organ discharge, the profile of the electric organ discharge is the same for the same fish, but it's a little bit different from one fish to the other. And this is enough for the wavelet to reveal that. You can use the wavelet, it's amazing. You can see some components of the wavelet. Exactly. 50 kilohertz. So we have 11 electrodes at 50 kilohertz. It's a huge amount of data, and that's why you're trying to do that in real time and record only which one is the fish and with the time of the firing. So at the first, when you let it learn and try to discriminate between fish, this is the result. It's really poor. Green is one fish and red is the other one. You have lots of noise here, lots of noise there. And especially these points here and those points here are the moments where both fish fire together. Then the wavelet and anything works good. So what we do is like smoothing. We look the previous spike of both fishes, and we say, okay, the fish cannot move too quick. So the spike now should be a linear combination of those two previous spikes moving in time. And then we choose which one fits better what we found here, and then we can discriminate like this. These points here are from the projector. Okay? So it's pretty cool. We can really separate them. Okay, and future. Well, for the future is those experiments he was saying, how we can try to figure out when the fish is trying to communicate or electrolocate. And we want to do chronic implant in the fishes and have the fish wireless sending this data to us and compare the behavior of these electrical signals when the fish is able to move with those data with the immobilized fish. Okay? I have 10 minutes more? 20 now, I think. 5 more minutes? Okay. Okay. This is really quick. What we don't know about infrared sensing in rattlesnakes. Well, this work was done in collaboration with the people in Rio Claro. Some biologists there that usually go to the field. They go to an island in the front of Santos that's called Queimada Grande. Yes, it's full of snakes. And it seems that there is some theory that it seems they call it Queimada Grande because there were lots of snakes there. Then they decide to pour gasoline or something there and put fire to kill the snakes. And instead of killing all the snakes, it seems that it selected only the most poisonous. It's something like that. Yes, because it seems that the poison is so concentrated that if they bite a bird or something, the bird cannot fly 2 meters and is dead. Because they need to survive, right? The bird needs to fall in the island. If the bird falls in the ocean, the snakes die, right? So it seems that they selected only the nasty snakes. We go there to dive, but we never go into the island. But these biologists go there. And one of them was talking to us that he figured out that whenever he found a rattlesnake in a tree or something, the rattlesnake was looking to the coldest region of the sea. It was something that they observed. And then they start, okay, well, it can be. I don't know. Then they bought an infrared camera, and then they start looking very carefully and not making noise for the snake. Don't realize that people are coming. And every time they found a rattlesnake looking to some region, they went there, they took out the rattlesnake, put the camera, and make a movie of and see that really the snake was looking for the coldest region. And then I ask why? Well, biologically thinking, there is a reason for that. If you have a prey that's a warm-blooded flying or walking around, and you have a cold background, you have a good distinction between them. So it's good for the snake. The snake knows exactly that there's something for eating there. And then they try to publish this with this data, with the camera and so on. And then they get the answer something like this. You need electrophysiological data to prove that. And then they came to us. Okay, can we record something? And then we start looking, how does it work? So it's something like this. The vipers and the rattlesnakes, they have this hole here that's called a pit, the L'Oreal pit. It's something close to the eye, but instead of having substances that are sensitive to electromagnetic spectrum in the visible region, they are sensitive in the region of the infrared. Okay, so it's a hole here with lots of infrared receptors inside. And it's called the L'Oreal pit. Fossetta L'Oreal. Well, the L'Oreal pit is here. This is the eye. You have the trigeminal nerve that innervate both of them. So the signal from the pit goes from the pit to the trigeminal nerve, gets mixed up with the vision signals that come here, and then it turns up to go to this region here that's called optical tectum. Okay? So this is the brain of the Crotalus, the rattlesnake. This is the optical tectum here. So if we stick an electrode here at the surface and you stimulate the eyes of the snake, and the surface of this region, you have most of the cells are sensitive to vision, to visual stimulation. Then you start going deeper and deeper and deeper. You find cells that are both sensitive to infrared and to visual stimulation. And deeper, very deep, you find cells that are really selective to only infrared stimulation. Like here, topographic. So you have both, for example, something that comes in this direction 1 and direction 1 in the pit, they come to the same region here in the optical tectum. But the visual stimulation is found more in the surface and the infrared more deeper.
Do you know if there's tecto-tecto inhibition?
It seems to be, yes. It seems to be, yes. Yes. Well, it's like this, right? So you have a sequence here, it's a little different, but they go to the same regions there. So from that, we believe that snake sees something like this. That's a mixed of, since the signals are going to the region where the vision is generated and also infrared, so they should see something that is a mixture between visual and infrared stimulation together. So they can see if there is some form there and where this is warmer and where it's colder. Okay? So that's the idea. Well, the experiment we did, this is the electromagnetic spectrum, just to explain a little bit how we physicists understand how the sensors are able to sense light. So the visible spectrum is here, the infrared is here. So if you have something like energy, the energy is bigger in this direction. So light photons have more energy than infrared photons and so on. And we believe that the cavity of where the sensors are is under thermal equilibrium, okay? And all the radiation that comes into this cavity is absorbed. Okay, this is what we call in physics a black body. Okay, so you have a black body with in thermal equilibrium. And when you choose this model to explain what's going on, you need to remember that this black body, for example, in our case, our body is kept in 36 Celsius. So our eye is like a black body in thermal equilibrium with an average temperature of 36 Celsius degrees. That means the cells of our black body are changing photons from each other, from inside, and every time from cell to cell. And the average energy of these photons is the constant of Boltzmann times the temperature, kBT. Okay? So these photons are more or less in this region, the infrared region. That's the thermal photons. Okay? So how we detect light? Light, the photons of light, they have a higher energy. We have a threshold of energy for our sensors that is higher than the near energy of the thermal photons. Okay? When a photon of red light, for example, comes, it has energy that's bigger than the threshold, so it's able to stimulate the receptor. And then we see red. Okay? So that's the idea. Well, and we believe, everybody believes that the infrared sense of the snakes should be the same. And that's inclusive, and that's actually the reason why the snakes are cold-blooded. And they try even to ventilate the L'Oreal pits to keep them colder than the environment. Right? What for? For the KBT photons of the L'Oreal pit that are black, it's a black body and are going in and out there, should be with less energy. They should have lower energy than the photons of infrared of the 36 degrees bodies that the snake wants to detect. Okay? That's the idea. Well, so that's the idea of the black body. You have the radiation that goes there, keeps there and never goes out. And you have a profile that's called the characteristic spectrum of emission of a black body. This is for a 25 degrees Celsius black body. And this region here, where you have the maximum absorbance or emittance, you have in between 5 and 15 micrometers. That's exactly the region where the photons of heat of a 30, 20 something, 28, 35 degrees are. So this explains that, okay, it seems to be a real black body because it absorbs better the region where the photons that we want to detect are. Okay. And, okay, the L'Oreal pit is always a little cooler, the no object to be seen, and we did the experiments. Well, they pick up the snake, they anesthetize the snake with isoflurane, right? Is this the name? Isoflurane. So the snake is sleeping, then they remove the teeth of the snake with just in case. They put this tube for respiration because you need to ventilate artificially the snake because with the isoflurane it stops breathing. And you need to put the isoflurane into the breathing too to keep the snake sleeping. And then they seal the mouth of the snake just in case the snake, something goes wrong and the snake starts to wake up in the middle of the experiment. It never happened. This is the L'Oreal pit. We can do experiments even in both ways. We can either cover the eye or we can cover the L'Oreal pit so we can know exactly what kind of stimulation we are doing to the snake. And then we open a little hole here in the skin of the snake. Here is the bone. We make a small hole in the bone with a dentist drill, and then we stick an electrode there. You need to be very careful because here there is an artery. If you touch the artery, oh, it's more thin. It's flexible. It's flexible. It's very thin. It's like 0.1 millimeters, something like that. It's very thin. No. Yes. Okay, then you have the electrode there, and then you can introduce the electrode deeper or more shallow to record the signals. So here is the snake in the table already with the fixes. We fix the skull. The problem is that everything, even the skull is kind of soft. It's hard to keep it together when you're trying to stimulate and put in the electrode. So we need to glue the skull to this piece of metal using this dentist cement. Try to glue it because if not, you start doing the experiment and then the head starts moving and then you lose the record. It's terrible. Well, and here is the little hole in the brain. and here is the electrode inside the region. Okay, so this is the snake in the apparatus, already with the microscope here, the electrode, and then we show the stimulus to the snake. How we build the stimulus is like this. So it's part of a sphere here, is a section of a sphere, okay? We have servo motors here that move this thing that we call prey, that we can control both the temperature of the background, fix it, and the prey. We call the prey because, okay, it's anesthetized snake with the prey moving around. Should be a really nice experiment for them. But okay, so this is the prey. Well, that was the first idea. We need to have something fixed, right? To quantify. So let's fix the background, let's fix the prey, the temperature, and then we move this prey around the regions of sensitivity. And then we look for a neuron that's responding to that. That's the idea. So this thing keeps doing like this, every 2 seconds. We put something like 22 degrees here and 36 degrees here, and that's it. We start looking for a neuron that responds to that. Well, what we found, the snake, these are one of the most interesting experiments we did. The snake was at 22 degrees Celsius, that was the air conditioning of the room. The background, we set it in 27 degrees Celsius, that's warmer than the snake. And we changed the temperature of the prey from 5 to 38 Celsius degrees. Oh? How many times for doing that? Well, from the beginning to the end, I think this is minutes. 700 minutes, 10 minutes. Okay, what we did was we freeze the prey, and then we applied the prey is a resistor. We freeze the resistor with one of these Freon coolant cleaning things, and it also has a thermal part there to measure the temperature. So we freeze it to 5 degrees, and then we put some very small current in the resistor and let it go to higher temperatures. And we record the temperature during the time, and the thing is moving. That was the idea, simplest idea to do. Well, I just showing from 0 difference to positive, what does it mean? This is minus 1.1. This means that when both background and the prey, here is the difference of temperature between prey and background. Okay? So this is when they are almost at the same temperature, and you start eating the prey. So at the beginning, when you have almost 0, you can see that no sensitive response. Oh, let me explain this. You can see a double blob here, right? What does it mean? This is time in seconds, from 0 to 2. The period of oscillation of the prey was 2 seconds. What does it mean? From 0 to 1 second, the prey is moving left to right. From 1 to 2, it's moving right to left. Okay, so it goes and comes back. Goes and comes back. Goes and comes back. So you see, goes and comes back. So this is the same region as this. Here is the temperature. Is this scale here. So here is almost 0. Here is 0.7 degrees, 1.1, 5.3. So here the prey is 5.3 warmer than the background. Here is 1.1, here is about 0. So when the prey and the background have the same temperature, it makes sense. The infrared sensor, just noise. Okay? When you start eating a little bit, like 1 degree already is enough to have a reliable response. When it passes to a same region, passing here, you can listen. You have to fish. You have to fish. What we did was we find one, we see if it's reliable, then we close the eye, open the pit, close the pit, open that, just to see if it's really very selective to to infrared sensing, and then we do the experiment. And then we move and look for another one.
What happens when you have a gray that is colder than the background?
Yeah, that's the white thing I'm going to show. Good question. Okay, so this is all we expected. So this is black body like. This is exactly what we believe. Okay? And what we believe that should happen below? Noise, 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 right? Because why? Why? Because if you have a black body and the cells of the pit are changing photons with energy kBt, they cannot detect photons with energy below kBt, right? Because they are already completely beautiful, right? This is beautiful. When the biologists saw that, wow, this explains how does the rattlesnake is able to prey frog when they are younger and then prey rat when they are adults. Because frog is not warm-blooded. Even they are colder than the environment, sometimes 4, 5 degrees. So this explains this thing. So you see, frogs are much colder. So the rattlesnake is able to see the frog. The frog appears here. Is absence. So everybody else is spiking in other regions, but not when you see the frog. I believe that should be some network effect. I don't know if it's at the level of the pit membrane or if it's somewhere else, but I think there is some processing that does that. And it has to do something to do with this ability to goes back to background. And it happens every time. The first experiments we did, we use iron for welding components, right? We turn on the iron, we put the snake there stuck and look into the iron, then we start doing like this with the iron in front of the snake, and then we look for answering the neurons. And what happens that when you know, okay, this neuron is sensitive to this angle, okay, to this section, and when you put the iron there, then you got lots of answer. If you leave the iron, background. And then you take out the iron. This is really cool. This first experience, we did some recording in the trigeminal nerve. So we are recording like, I don't know how many, 500, 1,000 fibers together. So you have lots of units there. So you put the iron, you get a signal like this size. Leave the iron, background. But background, you have some activity. Then you take out the iron, the background turns like this. Almost disappear, and then background again. And then we got a frozen snake that the guy had there. And then we show the frozen snake to the pit. And then background activity, frozen snake. Leave it there, background. Remove frozen snake, as if it was the iron. And then back to background. After 4 seconds, 3-4 seconds. Yeah, very, very quick. Yeah, but how to explain that without the KBT? Oh, this is interesting. That's exactly what I'm going to show here. So biological problem solved. But how to model it? And then we found this, nanopits. Some guys, the Spike Technologies, it's a company. They have a website or something. They published this paper, and they said that if you look at the membrane of the L'Oreal pit, actually you have, it's not a membrane. You have nanopits inside it.
So you need the photon to come from a direction, not from any.
Maybe.
That's why you need.
Maybe. And this could be, I don't know. We need to think about. Roland sat a little bit with this for some hours and told me, oh, I think I found it. And then it seems that the calculations didn't convert, so I don't know. But we don't know yet how it happens. Should be something related to connection, network effect, and geometry, but it's not easy. It's really interesting. And this is known. This kind of behavior is you find in textbooks since the 60s. I mean, it's 50 years ago. But nobody tried to do with colder temperatures. It's really amazing, right? And then when they put cold temperatures, our models just fail. It's really nice. Okay, so this is basically what I have to show you. Thank you very much. Huh? Yes, to leave the snakes awake is not a good. But eu tenho agradecimento para o Pedro. O Pedro que fez a primeira. O Pedro que abriu a primeira cobra. Ele que me ajudou a procurar o.